Hi guys, in this video, we're going to discuss the motor system and specifically how movement is controlled. So in short, movement is controlled by muscle contraction, which is mediated by filaments like actin and myosin, which are regulated by calcium ions. Before we talk about the cellular mechanism, let's talk about the motor system from a macro level. Let's look at a human body diagram. The motor neuron cell bodies are in the brainstem and spinal cord, and their axons travel to organs and muscles in the bodies and limbs. So like it goes to our arms and legs. From a macro level, movement control is hierarchical, as you can see from this relational diagram. The motor cortex sends information downstream to different parts of the brain, then to the motor neurons, and then finally to skeletal muscles. Notice that the motor system receives input from the environment through sensory neurons at multiple levels in this hierarchy, and also the loops that involves the basal ganglia and the cerebellum are important because they refine motor controls. So in the next 7 minutes or so, we're going to look into how filaments like actin and myosin and calcium ions mediate muscle contraction. Firstly, let's look at the muscle cell and its organization. So this long pink cylinder structure here is one muscle cell, and it contains multiple myofibrils. Each myofibril has a repeating unit called sarcomere. So each sarcomere consists parallel overlapping filaments. Filaments are thread-like, long chain structure of protein subunits. There are two kinds of filaments on the diagram on the right. The thin filaments are F-actin, shown in red. The thick filaments are myosin, shown in green. The movement of this filament is controlled by calcium ions, and they require ATPs to move. But we have to talk about the other structures in the muscle cell first to understand the whole mechanism. So now let's zoom out a little and see how everything holds up together in 3D. The sarcoplasmic reticulum is a network of tubules that extend throughout the muscle cell, wrapping around but not in direct contact with the myofibril. Another structure is transverse tubule, known as T-tubules, which are extensions of the cell membrane that brings plasma membrane close to the sarcoplasmic reticulum and travels into the center of the cell. So these are T-tubules, and then these like network structure are sarcoplasmic reticulum. So the functionality of sarcoplasmic reticulum is that it regulates the level of calcium through storing and releasing calcium ions, which is needed for muscle contraction. The T-tubules permit rapid transmission of the action potential into the cell and plays an important role in regulating cellular calcium concentration. So therefore, this structure allows calcium ions to be distributed quickly throughout the cytosol. So now let's talk about the role that calcium plays in muscle contraction. On the right is an image of neuromuscular junction, the interface between the nervous system and the muscle. We know that the arrival of an action potential will cause a release of acetylcholine at the synaptic cleft, which binds and opens the nicotinic acetylcholine receptor and cation channels at the neuromuscular junction. The binding of acetylcholine, like right here, will open the cation channels and causes an influx of sodium ions that depolarizes the muscle cell and produces action potentials within the muscle cell itself. So the T-tubules effectively propagates the action potential to the entire cell, and that just means that like it will just pass the action potentials like downstream. And remember, one functionality of the sarcoplasmic reticulum is storing and releasing calcium ions. The action potential causes depolarization in the muscle cell that opens voltage-dependent calcium channels, and calcium ions are released from sarcoplasmic reticulum. And remember, the T-tubules bring plasma membrane close to the sarcoplasmic reticulum, and it travels into the cell. So it can allow all sarcomeres in the same muscle cell to contract nearly all synchronously, which is an important property to have. So now let's also look at how the calcium ions interact with muscle filaments. Muscles contract by shortening the sarcomeres. So when the muscle is relaxed and under low calcium concentration, actin is coated by tropomyosin and troponin. So in the diagram on the left, you can see the pink circles are actin subunits, the purple line are tropomyosin, the blue boxes are troponin, and the red dots are calcium ions, and the green dots are myosin binding sites. So the myosin binding sites are blocked by tropomyosin when the muscle is in relaxed state. When calcium ions binds to tropomyosin, that means when the calcium concentration is high, then it will cause a conformational change in the troponin complex, displacing tropomyosin along the actin filaments and allow myosin to bind to actin that later produce a mechanical force. So therefore, a rise in calcium concentration caused by actin potentials will allow the binding of myosin to actin. So muscle contraction needs energy. So that's why we also need to talk about ATP. Myosin is a motor protein and it has another binding site for ATP, at which enzymatic activity hydrolyzes ATP to ADP, releasing an inorganic phosphate molecule and energy. So this is known as ATP hydrolysis. ATP binding causes myosin to release actin, allowing actin and myosin to detach from each other. After this happens, the newly bound ATP is converted to ADP and inorganic phosphate. The energy released during ATP hydrolysis changes the angle of the myosin head into a cocked position. So if the actin binding sites are uncovered, which they are, a cross bridge will form. That is, the myosin head spans the distance between the actin and the myosin molecule. The inorganic phosphate is then released, which reinforces the binding interaction between myosin and actin and subsequently triggers the power stroke. The power stroke is the key force generating step used by myosin. So forces are generated on the actin filaments as the myosin protein reverts back to its original conformation. The myosin head moves towards the center of the sarcomere. So these are the myosin heads. So these myosin heads will pull the actin along with it. So as the actin is pulled towards the center of the sarcomere shortens and the muscle contracts. So you can see this muscle contraction in the diagram on the right here. So this sarcomere shortens. So this is referred to as the sliding filament model because the filaments move towards the opposing directions, which looks like they are sliding into each other. So to summarize what we've talked about so far, an action potential from the presynaptic neuron will cause a release of acetylcholine that causes an influx of positive ions into the muscle cell. The muscle cell is then depolarized and an action potential is produced within the muscle cell itself. Calcium channels are open and calcium ions are released. And then they cause myosin to bind to actin through a conformational change. The myosin uses ATP to pull actin towards the middle of the sarcomere, thereby shortening the length of the sarcomere, which is known as muscle contraction.